Hello and welcome to our webinar series, Research Clues to Appreciating a New Era of Cannabinoid Conundrums. Our second webinar of the series will be presented by Dr. Svante Vikingson, Research Forensic Scientist with the National Laboratory Certification Program at RTI International. Without further delay, here is Dr. Vikingson.
Thank you for that introduction. I'm here to talk to you today about the drugs out there that walks and talks like cannabis, but actually aren't delta-9 THC. I'm going to try and consistently call them THC Isomers, but I've also seen THC analogs or semi-synthetic cannabinoids. And that last one is kind of fitting, given that they are sometimes produced by converting CBD extracted from hemp. In the next 35 to 40 minutes, we'll cover the nomenclature, prevalence, and effects of the THC isomers. And then we'll finish up by talking a bit about the analytical challenges associated with this. This webinar is part of the FTCOE, but I also like to acknowledge the support from SAMHSA through the NLCP program for most of the information I'll present today. So are THC isomers part of the NPS phenomenon? Today, I'm going to argue the point that they are similar and might require similar responses, but also that they are different beasts. As you will see in my talk, there are important similarities. They are minor modifications of classic drugs, in this case, cannabis. The new analogs are introduced regularly, and we know very little about their effects. But there are also some very important differences. The THC analogs are sold much more openly, and they have a much larger user base that is also very different from the users of other NPS, and they are typically produced from hemp, which I'm come back to why it's important. So in summary, my personal opinion is that this is a different phenomenon from NPS, but also one that we must take seriously and which might be a bigger concern when it comes to workplace and road safety. So let's jump in feet first at the deep end and take a look at the different isomers. The THC molecule can be modified in different ways, the most important being changing the position of the double bond and create new homologs. As you can see on the top of this slide, the double bond can be anywhere in the ring and on the methyl group there on the top. In addition, the double bond can be removed altogether, creating hexahydrocannabinol or HHC. Of these, the four grayed out are less common, and my suspicion is that we're never going to see them as major cannabinoids in any product. And the reason for this is pretty simple. Most of these are in one way or another produced from CBD extracted from hemp. And while the synthesis of drugs like delta-8 or HHC is fairly simple, the synthesis of delta-7 and delta-7,6A7 are very complex. Delta-911, also known as exo-THC, caught people's attention for a while, but is actually more of a byproduct from delta-8 synthesis than a drug of its own. Another important group are the side chain homologs. On the bottom there, we have THCB, THCH, and THCP, which has side chains of 4, 6, and 7 carbons respectively. And these have all been marketed. Well, the one on the far right there, marketed as a THCJD or the THC octyl homolog, it appears that those products might have been fake, for a better word, and might have contained a different isomers. While at least some of the homologs are present in cannabis, there's typically very little of them, and as far as I know, it's very difficult to change that chain length in the final extract. And I've seen many examples of manufacturers highlighting analogs such as THCP in marketing of full spectrum blend, even though the bulk of the cannabinoids is something else. And when they are sold pure, the homologs are likely synthesized from non-hemp precursors, contrary to what the producers claim. The homologs are also found in delta-8 variants, where the double bond has moved to the 8th position, which actually might be the most common version potentially as delta 8 is more stable than the delta 9. It doesn't end with the double bond modifications though. We also have the conjugates such as THC-O acetate, also known as THC-O. And this molecule should not be confused with THC-A, which is a major cannabinoid found in the plant that breaks down to THC when heated. As you can see here, they are both acetates, but in THC-A, the acetate is actually in a different position. And if we then look at the bottom here, we can see that the producers weren't content with modifying only one part of the molecule at the time, and we are starting to see combinations. I've highlighted 3 THC Isomers that I recently saw marketed. And before we move on from looking at these structures, I also want you to know that even though we're talking about the THC Isomers today, there's also CBD isomers out there now. And several of the modifications that we see for THC isomers has also been applied to the CBD molecule. If you want to dig deeper into the THC isomers and how they are produced, I'd highly recommend the 2 talks by Jeffrey Williams of Cayman Chemical that were given during the 2023 Recent Trends in Forensic Toxicology for a more detailed look on especially the synthesis of these molecules. So moving on to discuss how big of a problem is this and how common are the THC isomers. It can be very hard to get a feel for the prevalence as the isomers don't kill too many people, frankly, and many of the forensic labs don't measure them. And part of the reason for this is that prosecutors are unable or unwilling to pursue them under the various state laws. And we can get a sense for the delta 8 prevalence by looking at workplace drug testing data. Delta 8 THC cross-reacts very well with the immunoassays used in our laboratories. And in a post-testing study conducted in the second half of 2022, we observed delta 8 in around 30% of specimens screening positive for cannabinoids. Now, delta 8 also occurs naturally in cannabis, which complicates the estimate. But to make a long story short, I estimate that the use of delta 8 THC is around 27% of the use of delta 9 THC, based on the relative prevalence of delta 8 and delta 9 THC carboxy in urine specimens. And you can see the distribution here in that graph, where the bar on the far left there is cannabis use. So this number is significant, and it makes the delta 8 THC one of the most prevalent drugs in our program, and that's important to remember. So data on the other analogs is less complete and less convincing, but still very interesting. So if we look at seized drug data from last year, we can see that delta 10 THC started to increase in October of 21. That's the graph on the top there. And in end-use data, which is a web surveillance program, they showed that HHC shatter started to increase around the same time. Apart from that, we do see many of these THC analogs, such as THC-O, HHC-O, and THC-P, being sold and marketed on various online websites, but it's a bit too early to tell if they will fade away again or if they will continue to become more prevalent. And how does this compare to the other NPS? Well, in my opinion, the THC isomer prevalence is of a different magnitude. Again, looking at data from the end use and their web surveillance, from time to time they publish alerts of drugs based on web chatter on drug user forums. I compiled the highest number of mentions in some of those reports for 2 THC isomers and 4 other NPS. And as you can see, the scale is very different. This is not a direct measure of prevalence, and we need to be careful. I did mention that the user base is different for THC isomers compared to the other NPS, and there might also be a difference in how they use web forums. So that could also skew this comparison. So if we accept the paradigm that THC isomers are very popular compared to most other NPS, the next question is why? We believe the most important factor is that the distributors and the users both believe this is legal. This ensures that the isomers, mainly Delta 8 at this point, are readily available and might also lower the barrier for using the drugs and reach individuals that would not use illegal drugs. Most of the Delta 8 is likely produced from CBD, and we believe that another driver for the THC isomer production is an overproduction of CBD that has led to a surplus of product that is hard to sell. On a related note, it's possible that some of the newer isomers, such as Delta 10 and HHC, are produced in an attempt to diversify and create a unique product. They are often described as having different highs compared to Delta 8 and Delta 9. And a third reason to use THC isomers could be to avoid detection in workplace or clinical drug testing. These reasons contribute to a THC isomer market, even in states with recreational marijuana. And another contributing factor there is the significant tax burden on marijuana that could make the THC isomers a cheaper alternative. And as a consequence, some states have prohibited the sale of especially Delta 8, which might be yet another driver behind the recent diversification in the THC isomers. As you can see, this marketplace works differently from that of other NPS. As such, I think we should think about the THC analogs as a separate problem. I also think we should be aware that when new analogs come to market, the industry will use the distribution network already in place, and we might see the prevalence rise very rapidly. Before we move on, I would like to touch on the issue of manufacturing. While this has nothing to do with the properties of the drug itself, it is still a concern. If the users don't know what they are buying, it will increase the risk of accidents. And the concerns can mainly be divided into three different categories. First, the concentration of, for example, Delta 8 THC might not be what is on the label. Cannabinoids are very difficult to formulate due to limited solubility and a tendency to get attached to surfaces. I have noticed that often the certificate of analysis presented by the vendor is not for the final product, but for the cannabinoid concentrate used to prepare it. Second, the products might contain more Delta 9 THC than advertised, in some cases more than what is allowed by the Farm Bill. This is an obvious concern, both with regards to the legality, but also for users who might surprisingly test positive on a workplace drug test. And third, there is a concern with other chemicals. Delta 8 THC is produced from CBD using a chemical reaction, and there is a risk that solvents from that reaction end up in the product. There is also a lack of product testing for things like pesticides, heavy metals, and microbials. And working with a concentrate from a plant, there is always a risk that unwanted components accumulate. A couple of organizations have done product testing studies lately, and even though the products weren't horrendous, they did not reach the level we have come to expect from a grocery store either. Many products contained too much Delta 9 THC, and some were also high on the metal content. And with regards to Delta 8, most products were over-labeled. That is, that the concentrations were lower than what's on the label, which to me is less of a concern than the opposite, but I'm sure many buyers disagree with me on that. So I said that the users believe that the Delta 8 THC and other isomers are legal, but are they? The short answer is probably yes. In a recent trademark infringement case, a U.S. appellation court held that Delta 8 THC was in fact legal under the current framework, which probably is the best guidance we have at the moment. The longer answer to the question is complex. The Farm Bill has made the claim that CBD and all analogs present in the plant are legal, but what it really says is that cannabis with less than 0.3% of Delta 9 THC by dry weight is hemp and therefore legal, And products made from this material is also legal as long as the delta-9 concentration does not go above 0.3% by dry weight. However, and here's where it gets complicated, the bill also states that all synthetically derived cannabinoids are still Schedule 1. And this is the critical point. There is not enough delta-8 THC in cannabis to make it profitable to extract it. Instead, CBD is extracted and converted to delta-8 through a chemical reaction. If this is considered synthetically derived or not, might determine the legal status of delta-8 THC and potentially other analogs with similar production methods. Another legal aspect often overlooked is that while being legal under the Farm Bill, products made for human ingestion or topical application also requires FDA approval. And FDA are more clear and state that the delta-8 THC has not been approved by FDA for safe use in any context, and in addition, it is not generally regarded as safe or GRAS. The implication of this is that most, if not all, products with THC isomers are still illegal to market for human consumption, even though the drug itself is legal. And speaking of the FDA, they actually went a step further and put out a 5-bullet warning about delta-8. They note that the National Poison Control Centers have received over 2,000 cases, of which 41% involved pediatric patients, and most of them were unintentional exposures. The number of children unintentionally ingesting these drugs is scary, and exactly why packaging is a problem. There is a reason why you cannot buy Tylenol gummies looking like a punny copy of the Sour Patch Kids. So 8% of the cases were admitted to a critical care unit, and in one case was coded as outcome death. And the discussion about the lethality of cannabinoids is for a different day. But the take-home message is that delta-8 THC, and I think the other isomers as well, are not necessarily as benign as the producers make them out to be. The FDA also point out the risks of harmful chemicals in delta-8 products, and that delta-8 THC will make you high. And that is what we're going to talk about next. So how impaired will you be if you drive after using delta-8 THC, and in what way? As you will see throughout the next few slides, most research in this area is focused on comparing the effects and potency of the isomers to that of delta-9 THC, and I think that's a good approach. In this recent dosing study, the effects of delta-8 were compared to 2 doses of delta-9 in 3 individuals, and the authors show similar kinetics and similar type and duration of impairment, measured as self-assessment and through horizontal gaze nystagmus, or HGN. Unfortunately, there's a couple of things that prevent us from using this research to estimate the potency of delta-8. First, the mode of administration is different. The authors compare smoked delta-9 to vape delta-8. And secondly, the products used were not tested by the authors to ensure the correct doses. And third, HGN is not a common measure of cannabis impairment. And in fact, it has traditionally been viewed as more of a sign of impairment from sedatives such as alcohol, and the presence of after cannabis use is controversial. That said, the results are well in line with what we expect from delta-8, and it's still one of few studies conducted. The 2 other studies I could find were both published in 1973 and are similarly terse in their description of methods and results. They both report similar effects between delta-9 and delta-8 THC, but that the delta-8 has a lower potency. One study mentions 2/3. So if we summarize the 3 studies on delta-8, we still lack evidence to say for sure, but it looks like delta-8 produces the same type of impairment as delta-9, and potentially it's slightly less potent. I'm proud to say that we are in the process of rectifying this. conducting a delta-8 dosing study in collaboration with Johns Hopkins University that we hope will be presented in the next few months. For delta-10 THC, there's a single study in pigeons that didn't report any effects similar to delta-9. This does not match the user reports and other anecdotal evidence out there, which indicate that it is active similar to delta-9. My guess is that this might be connected to chirality. All cannabinoids have chiral centers and produce enantiomers with varying efficacies. Enantiomers would be the mirror images. Now, delta-10 THC has 2 chiral centers and can therefore exist in 4 different forms. It is possible that the study was carried out with an inactive form, and in addition, I must confess that I'm not familiar with the pigeon as an animal model, which could also have contributed to these results. For HHC, there is one study in mice showing various effects and a potency 7 to 34 times less than delta-9. The study was carried out with a mixture of the enantiomers, and one of them has been shown to be inactive by computer modeling, indicating that the active enantiomer might be twice as potent as indicated by the results. And in THC, for THC-O, it's reported to be more potent than corresponding THC. And my belief is that the effects of THC-O comes from a conversion to THC, and a higher potency can then be explained by improved pharmacokinetics. For example, perhaps THC-O can cross membranes easier since it's slightly less polar. We don't really know yet. And for these less prevalent isomers, the studies indicate that most, if not all, of the isomers produce effects similar to delta-9 THC, but with varying potency. The studies on this page is actually a pretty good representation of another important point here. The evidence is made up of a mixture of different types of studies, making it difficult both to compare and estimate the relevance for impairment while driving. And I just picked one of the studies here to illustrate some of the difficulty in interpreting these studies. And that's a study by Martin et al., who studied delta-8 cannabinoids with different chain lengths from 5 to 8 carbons. In general, the potency seems to increase with increasing chain length, at least up to the 8 carbons in THC octyl or JD. And the bar chart shows the potency relative to delta-8 THC at different chain lengths in 4 different tests. The green bars are a protein binding assay, measuring how hard the analogs bind to different cannabinoid receptors. The blue bars represent potency in 3 different measures of effect. While we see a general trend, there are major differences between the tests. And for example, THC-H was most potent with regards to body temperature, and THC-P most potent with regards to spontaneous activity. The take home is to approach potency determinations with caution and a grain of salt, especially with regards to in vitro studies that are conducted in animal models, and when we only have data from a single assay. Yep, I know that pretty much put all of the evidence we've seen so far into question, but for these isomers, the evidence level is just too low. And while we're waiting for more evidence to come in, I think we should consider these isomers all being impairing. THC isomers are also challenging from an analytical standpoint, and we'll round this webinar up by looking at some of them at a conceptual level. Roughly, the challenges can be divided into 3 categories. First, they cross-react in immunoassays and therefore generate screening positive specimens that the laboratory have to analyze with a confirmation method before reporting as negative, which can be burdensome. Second, the THC isomers can also interfere in chromatographic methods, particularly in LC-MS. By eluding close to the analyte of interest and producing unreportable peak shapes and/or ion ratios. This is concerning as it allows for marijuana use to go unreported. And thirdly, when we are looking for the THC isomers itself, they can be difficult to detect as we know very little about their metabolism. And this is especially true for urine, where we know that THC is normally detected as a carboxy metabolite with only small amounts present of the parent drug. While it's assumed that that is true also for THC isomers, we don't know this for sure, and very few reference materials exist for those metabolites. So, looking a little bit closer on the immunoassay screening, marijuana use is often detected using assays targeted the carboxylated metabolites of delta-9 THC. Our studies show that the carboxylated metabolites of delta-8 carboxy cross-react very well with those immunoassays, and we conducted 2 experiments to confirm this. In the first experiment, we asked our 24 urine drug testing labs to compare the signals for 100 nanograms per milliliter of delta-8 carboxy and the same concentration of delta-9 carboxy. And it turns out that the signals were very close, with the delta-8 carboxy signaling averaging 99% that of delta-9 carboxy. However, there was a concern that since this concentration is twice the cutoff, that there would be an issue with signal saturation. So we did a second study in which we asked 3 labs to use different assays to test a series of concentrations around the relevant immunoassay cutoffs. And you can see that on the right there. And at 50 nanogram per milliliter, the cross-reactivity was between 87 and 112% in the 3 assays. And if you're more of a visual person, that would be the concentration where the red line crosses the horizontal black line in these graphs. And the implication of this is that delta-8 THC carboxy can be very well detected in these assays. For whole blood ELISAs, Moody et al. Reported a similarly high cross-reactivity, very little for delta-8. Very little is known about the cross-reactivity of the other THC isomers, simply because there just aren't any reference materials available yet for the urinary metabolites. In the Moody study, 7 to 25% cross-reactivity for the parent compounds XO-THC, delta-10 THC, and delta-6A-10A THC was reported, which we can compare to 22% and 45% for the delta-9 and delta-8 THC parent compounds, according to the product insert. And given the similar cross-reactivities for the parent compound, there is hope that the carboxylic acid metabolites, if present, could cross-react quite well in this assay, but we still need to verify this. They also reported a lower cross-reactivity for THC-P and THC-O, which would indicate that those regions are more important for assay recognition. If I dare to speculate, I'd say it also implies that sensitivity for metabolites of the homologs and pre-drugs could be lower. So now we have a cannabinoid presumptive positive, and it's time to confirm it, which we usually do either with GCMS or LCMS-MS. Here, the structural similarity between the THC isomers and the delta-9 THC are making them very difficult to separate chromatographically. And please remember that this does not only apply to the parent analyte, but also to the metabolite, and it applies even to metabolites for which there are no reference materials, making this more difficult. And given the prevalence, delta-8 THC is the big issue, and that issue looks a little bit different if we look at LCMS or GCMS. In LCMS, delta-9 and delta-8 THC behave very similarly in the mass spec, often producing similar ion ratios. As you can see in the table here, delta-8 and delta-9 carboxy produced peaks with the same transitions and with almost identical ion ratios. In addition, in many chromatographies, we see a complete peak overlap, making it impossible to differentiate them. We're not going to go into detail on the different solutions, but let's just say there is no magic bullet, and laboratories are still struggling with balancing runtime and separation. Many laboratories have increased their runtimes and changing up their columns and mobile phases to increase this separation, while others are just embracing the similarity between the analytes and have instead dropped the delta designation and just report them together as just THC. We instead look at GCMS, delta-8 THC and delta-9 THC are typically better separated, even though the separation might be inferior in methods with very short runtimes. And as shown in the GCMS spectra below on the bottom here, the ions are still the same, but the relative intensity of the ions might be a little bit different between delta-8 and delta-9. And if the separation is not adequate, what happens is that a mixture of these two analytes will cause ion ratio failures and make it impossible to report either analyte as positive. To study this a little bit further, the NLCP conducted a special round of proficiency testing, challenging the 24 labs with various mixes of delta-8 and delta-9 carboxy. Most of the labs did great, but 3 labs that were using GCMS were unable to report delta-9 THC when the delta-8 THC ratio reached 10%, 20%, and 70% respectively. They were able to amend their methods and can now sufficiently separate the two isomers. And to me, this is an example of a perfectly good method requiring modification because the pattern of drug use and therefore the composition of the specimen changed over time. So if you thought that delta-8 THC was the only thing you need to worry about, I'm sorry to tell you that you might be wrong about that. Edward Sumida of the Palm Beach County Sheriff's Office reported that there is a risk of both delta-6A10A and exo-THC interfering with delta-9 THC in the different assays. And it's also likely that other THC isomers and their metabolites might also be interfering. And on this end, note the value of a good chromatographic separation cannot be overstated. I would also like to mention CBD. Again, not part of this talk, but I think I would like to make you all aware that CBD can convert to delta THC during sample prep. This can happen under acidic conditions, and especially if acidic derivatization agents are used before GCMS analysis. And this can be especially problematic if CBD is not measured and reported in the method, as the concentration of CBD and metabolites can be substantial. And even a small conversion can cause a negative specimen to become positive. If you haven't tested your methods for conversion, I strongly encourage you to do so. So as I mentioned before, we don't really have the time today for a complete review of methods capable of separating THC isomers. So instead, I'll show you these chromatograms from the development of a screening methods. Why do I do this? Because I wanted to send you along with a tip for improving the separation. I've seen a lot of examples of labs working on their separations by increasing the runtime or tweaking the gradient elution profile. This is tempting as it's easy to do and requires minimal changes to the laboratory workflow. However, a much more powerful tool to affect the separation is really the selectivity. So I urge you to get out there and try different columns and mobile phases. And the chromatograms here show the importance of column chemistry. The 4 colored chromatograms on the bottom are all from different C18 columns. And as you can see, the peaks all elute in pretty much the same order and don't change much. On the other hand, the top chromatogram is instead from a biphenyl column, and here you can see a different pattern with a different elution order. So to solve this conundrum, and indeed any separation problem, I would suggest starting out by running scouting gradients with different mobile phases on as many column chemistries as you have in the lab. And if you find something that works, share it with the rest of us, perhaps as an abstract to AFS or SOFT in 2024. So our final analytical challenge is sort of backwards compared to the others and come down to our lack of knowledge preventing us to identify the THC isomers when we are looking for them. The place where this becomes really important is when we rely on the metabolites to identify drug use, which is true for most drugs in urine, including delta-9 and delta-8, where the carboxy metabolites are the most important. Do we find either THC isomers as parent drug in urine? We don't know, but I would guess that chances are that they are also heavily metabolized. But then again, we don't even know what the major metabolites of the THC isomers are. The NLCP program recently synthesized delta-10 THC carboxy, but we have so far been unable to identify that metabolite in urine specimens, even though delta-10 is being sold. Could it be that another metabolite is the major urinary metabolite of delta-10 THC? Maybe. And to fill this knowledge gap, we need to do more research studies, ideally including both in vitro and in vivo studies. Using a mouse model or something like human liver microsomes might not be enough, though. The metabolism in animals can be different from humans. And since the microsome is not a complete body, the metabolic system is not complete either, which can skew what metabolites are found. So ideally, metabolite markers from those kind of investigations should be verified in dosed volunteers or in authentic specimens. And a special case here are the THC acetates, or THC-O, which are believed to work as cannabinoid prodrugs. We think they are readily degraded to THC in the body and then metabolites from there. However, if the degradation is complete, all we would find in the urine would be THC metabolites. And how do we differentiate use of the acetates compared to the parent drugs? I honestly don't know how we're going to go about that. So I'm kind of hoping that we'll eventually be able to find some sort of marker. And after listening to me for almost 40 minutes, there are a few things I'd like you to bring with you from this talk. First, that there are more THC isomers out there than delta-8, and we'll probably see this market evolve a bit more in the next couple of years. Second, THC isomers seem to produce a similar type of impairment as marijuana, even though the potency can be both lower or higher. And even though we are lacking most of the data we need, it's likely that the type of impairment is similar to marijuana, making the isomers a similar threat to workplace and traffic safety. And the drugs are perceived as legal, which affects both how they are sold and who buys them. Our data indicate that the prevalence of delta-8 THC is already one of the highest among drugs of abuse. Finally, there are analytical challenges with THC isomers, which can be divided into immunoassay cross-reactivity, interference in chromatographic assays, and lack of knowledge about the metabolites, making it difficult to detect immune matrices such as urine, where we depend on the metabolites for identification. And I also, again, want to make a strong case for ensuring that you have a good chromatographic separation. Before I let you go, I want to highlight the NLCP newsletter that comes out a few times a year that's called Drug Testing Matters, which articles on topics related to forensic toxicology. There are especially two articles that touches upon the topics of this presentation. First, an article on the effects of delta-8 THC interference in cannabinoid assays, and another on analytical challenges of cannabinoid metabolites, including conversion of CBD to THC. And both of them are written by our own Dale Hart, and I found them to be very interesting reads. If you're interested in these articles or articles on other topics in forensic toxicology, please use the QR code here for a link to our SharePoint site, or send me an email and I can direct you further. With that, I want to thank you for your attention, and I am ready to answer some of your questions. Thank you.
Hello, I'm Dr. Ruth Winokur, a principal scientist with the Center for Forensic Sciences Advancement and Application at RTI International. Thank you, Dr. Vikinson, for providing such an informative presentation. We will now begin the Q&A session. So first question, are there any plans within the USDA to mandate contaminant/potency testing for hemp grown for cannabinoids, just like every state requires for cannabis?
I do believe that I just looked in the USDA final rule from 2021, and it does have requirements for testing in there, but that would be for the hemp crop, not necessarily any consumer products. And I do think, you know, that that there is a need to test the consumer products, if that will be on a state basis or through the FDA or through the DEA. We'll have to see. I don't know more than anybody else at this point.
So the next question, a very good question, I think, and one that we recently talked about at a recent site visit with Samson. How is it possible for a forensic laboratory to prove something is synthetically derived? And has this come up in court?
That is the one, a very important question. I agree, and I think that's really where we're seeing this. Is, you know, one part of the argument is that since all of these many of these THC isomers, as I talked about, are natural to the plant at very, very low levels, it's clearly so that it's not commercially viable to extract them and make products. But since they, in theory, could be extracted rather than synthesized, that makes it very hard to discuss in court. So I don't think that has specifically come up. I think there is some work out there that's very promising, looking at, because we know quite well at this point what kind of processes are used to convert the CBD into these isomers. And there are some byproducts that are produced, and I think a very, I do think that those byproducts could be identified in products. If they can be identified in biological fluids or not, we still don't know, or what happens with them in biological fluids. But I do think that we might see markers of the synthesis route that was used, or the conversion process that was used, that we can detect, and those could evolve to become markers of that the product is synthetically derived. I think there are some initial work being done in that area.
Yeah, I think the most promising potential marker is the ISO delta 8. And that's been reported by Cayman, I believe. Yes,
I believe that is also mentioned in those webinars that I was referring to when we talked about the conversion earlier.
Mm-hmm. Great. Has there been any delta 8 combined with fentanyl studies published?
Not that I know of. I mean, I'm very sure they are used by the same individuals, because they're both very prevalent in our drug supply, and we do see a large amount of polydrug use in our drug using populations. But I have not seen any studies on the interactions between them, or on the on any particular that they would be associated somehow.
So the next question is about reporting. So in a response to the question regarding a forensic laboratory's ability to prove a substance is synthetically derived, do you report the THC isomers with the associated legal schedule on your reports?
Yeah, so well, first of all, I don't really report anything in my daily work. I'm privileged to work at an organization and the NLCP product that doesn't, we don't directly analyze specimens, but we work with the laboratories who do. And I do think it's a good question. I think I would say if your jurisdiction allows, it would be better to talk about delta 8 as being impairing, and try to avoid putting it in a specific schedule right now. because the situation is fluent where it is right now, and I think it might also change. So I guess that is my answer, and I also know that all laboratories are not even bothering to separate out and report delta 9 and delta 8 differently. And I'm also sure that in other jurisdictions, for some specimen types such as DUID, where you have a list of analytes that you can test for, that might be on a state basis, for example, that many of them probably don't contain delta 8 at this point.
Yeah, it's a particularly good conundrum for us, because the federal program that the NLCP oversees can only test for Schedule 1 and 2 substances, and so this delta 8 of whether or not it's a Schedule 1 or not is a conundrum for us. So, all right.
Indeed.
So, yeah. So we have a comment here, a fine point here, that the Farm Bill does not specifically state that synthetic hemp derivative components remain controlled. It is the DEA and their interim final rule that seeks to clarify this distinction in the definition of hemp, and by extension, tetrahydrocannabinols.
From what the commenter is writing, I am pretty sure that the commenter is more well versed in this particular legislation than I am, and I do think that an underlying problem here is that I'm not entirely sure that the delta 8s were something that the legislators foresaw happening when they made the Farm Bill legislation, which has sort of prompted other agencies to update and further clarify.
Yep. The next question is, are there any other related compounds with known conversion issues? And I'm going to assume we're talking about THC related compounds. Yes,
I'm assuming that too. Otherwise, we could spend the rest of our time talking about that. So if we're talking conversion in the laboratory, as far as I know, it is the cyclization of CBD to THC that has been published. I would say that I wouldn't be surprised if we see similar issues with THC carboxy and THC hydroxy as well. I don't see why we wouldn't have that same problem there, but it's not something I've really seen published.
Yeah. So the next one is interesting because we've also studied this. So despite one study by a cannabis company nearly a decade ago showing CBD can convert to THC in simulated stomach acid, no study has ever replicated those results. You mentioned CBD converting in sample prep in a lab. Are you aware of any valid studies showing that can happen in weaker stomach acids?
No. The short answer to that question is no. We do not believe it can. The slightly longer answer is that in the particular study, the Merrick study that showed the conversion, they had a huge problem with solubility of cannabinoids that I'm sure everyone who has worked with cannabinoids can relate to. And what they did was they added SDS as a solubilizer, and that we have in our own laboratory, as you point out, Ruth, studied this, and what we saw the same thing as we saw huge conversion in artificial gastric fluid when we added the SDS to keep it in solution, but we didn't see virtually any conversion at all if we remove the SDS. And so I think that part of that protocol is, the reason why they saw it. There are a number of human dosing studies that have dosed individuals orally with pure CBD without any THC in it, and do not see any conversion to measurable concentrations of THC. There are other studies out there that has sort of discussed the in vitro results and repeated some of the in vitro results. We have repeated those studies in our laboratory, and we don't see any conversion to speak of. When we're using CBD products that have an emulsifier in it, we do see a tiny little bit of conversion, but nothing that would lead to a drug effect or that could cause you to test positive for THC. The one caveat to that, though, is just because you have an individual who says they've used the CBD product, that doesn't mean they can't test positive for THC, because we do see a labeling issue with THC or with CBD products, as I believe we talked about in the earlier webinar in this series, where there might be unlabeled, or it might not be on the product that they does contain some trace concentration of THC, and even product with much lower than the 0.03% THC limit can give you a positive urine test for THC.
So the next question is about differences between human and mouse lungs. So as mouse lungs are radically different than human lungs, different amounts of lobes in each lung, among other major structural changes, can we actually rely on mouse models for inhaled cannabis products?
I agree that we cannot. I mean, it is, as I mentioned, it's tough to rely on mouse models or laboratory-based models. I was talking mainly about kinetics here, but I would say it's the same for drug effects or for impairment, that we can use the animal models when we don't have human studies to rely on, and they can give us a hint. But to really know what's going on, and especially if we get into things like trying to compare how potent the different THC isomers are to each other, I don't think the mouse models are good enough. We need dosing studies, and we need them to be controlled, so we know what products are used and what those products contain. The reason why we see so many animal models and so many binding assays and things like this is that to do these studies in humans is complicated and expensive when we're talking about new drugs of abuse that potentially could be Schedule 1. You have to design the study to use a brand new product that's a Schedule 1 in humans, and there's significant risk with that that you have to address, and that becomes complicated and costly.
Yeah, exactly. We know how expensive these dosing studies are. So, all right. So if CBD can be converted to Delta 8 THC and further converted to Delta 9, why stop at Delta 8? Why not go all the way to Delta 9?
That's an excellent question, and I think some of the producers have asked themselves the same question and started producing hemp-derived Delta 9 THC, arguing that since it's derived from CBD from a hemp plant, it should not be, it's not a scheduled drug because the THC doesn't actually count. I think that's a much weaker argument than for Delta 8, personally. And I do think that's really the answer to the question, is that individuals want to avoid the Delta 9 for fear of, it's legal issues, that it might be harder to sell. They might take a larger legal risk. There's detection issues. Some individuals just want to avoid using the drugs that they know their employer or their probation officer will be testing them for. And then switching to a different THC isomer might be a way to try and avoid detection. Thirdly, I think it's about preference, and a lot of these products are marketed as having differential effects, and if it's Delta 9 or Delta 8 or THC-O, but it could also be if it's terpenes and other additives, and I just think it's also product differentiation. You create something unique that people want to use.
Our next up is a comment. The commenter has reservations about THC-O being 15 times more potent as THC. They say the study was done in the early 1940s when the correct structure of Delta 9 THC was not known.
Yeah, I'm happy to agree, and I look forward to when we'll start to see some data coming out on that. As I said, it's mainly believed that THC-O has its effect by conversion to or breakdown to THC, and it's a little odd that it's that much more potent. I agree, and we are facing that, and a lot of these studies are very old. A lot of these studies are more than 50 years old, and the standards for research were completely different. So I'm completely agreeing. I've just tried to show the data I could find, whatever it was, and I am looking forward to see if modern data sort of showed the same thing.
All right. Well, unfortunately, we've run out of time for all audience questions, but please feel free to reach out to our presenter for additional information, or if we did not have time to answer your question. Thank you so much for attending, and thank you, Dr. Vikingson, for being here with us today.
Before you leave today's webinar, I'd like to draw your attention to our feedback form. It takes less than 5 minutes to complete and can be accessed via the checkbox icon directly above the chat. Your honest feedback is invaluable as we continually search for ways to improve and provide the best experience possible. Lastly, across the top of the live stream window are links to our social media profiles, including Twitter, LinkedIn, and Facebook. There is also a link to the FTCOE website, the Just Science podcast, and the FTCOE newsletter registration. Like, follow, or subscribe to stay up to date with everything the FTCOE is doing in support of the forensic community. Thank you again for attending, and we hope everyone has a great day.